So welcome to our second Anne Klibanski visiting lecture. I'm Miriam Bradella, the director of the Center for Faculty Development. And after our very successful kickoff of the series 2 weeks ago, I'm very excited that we have a great turnout today. And because of this, the goal of this lecture is really to advance women faculty across the MGH, the US, and the world. And before we start with our main lecture, I would like to briefly say a few words about the origin of this lecture series. So the Anne Klibanski Visiting Scholar Award was established because as we all know, women are hit disproportionately by the COVID-19 pandemic with added childcare and household responsibilities. But even before COVID, women lagged behind men in academic promotions and leadership positions. And one reason for the lower promotion rates of women are difficulties to travel for national and international presentations because often of childcare and household responsibilities. And it's not easy to leave your children behind to travel for a week for an international talk. But it is not just the line in your CV that you gave a talk, it also decreases opportunities to network and make connections and seek outside mentors and collaborators, which are all really critical to career advancement. Now with the COVID-19 related travel bans, most grant rounds and visiting professorships around the country are being conducted virtually. And this provides an opportunity to promote the participation of more women in visiting professorships. And you can now give an international talk from the comforts of your living room, as we will see or hear today. So the Center for Faculty Development used the silver lining of the pandemic to implement the Anne Klibanski Visiting Scholars Award. So we selected 36 women faculty, and they are clinicians, educators, researchers, and also postdocs at MGH who have shown exceptional promise as leaders in their field and whose careers would specifically benefit from speaking and mentoring and networking opportunities at a national or international level. And these women serve as virtual visiting professors and give grant rounds at a national or international institutions organized by the CFD. And we also identify a mentor at the host institutions. And then the scholars receive professional coaching and are part of a career development seminar series. A couple of words about Anne Klibanski, after whom the award was named. Dr. Klibanski is the current president and CEO of Mass General Brigham and the former director of the Center for Faculty Development, so my predecessor. And she has been a strong supporter and advocate for women physicians and scientists at MGH, including myself. Among many other initiatives, she was instrumental in establishing the MGH Backup Childcare Center and the Cleveland Distinguished Scholar Awards, which support the careers of female scientists at MGH at a critical period in their careers. And before Anne became the CEO, she was the chief academic officer at MGB and then the head of the neuroendocrine unit and extremely accomplished NIH-funded researchers with leadership roles at Harvard Catalyst, the Endocrine Society, and the NIH. So here is the picture of our amazing Anne Klibanski scholars that were selected. And here are some of the institutions where they're giving their presentations. So in order to promote as many women as possible, we thought that we should return the favor and host women faculty from the host institution to give a talk at MGH. So the Anne Klibanski Visiting Lecture Series was created to support and advance the careers of women faculty at MGH, the US, and also abroad. And these lectures now bring together faculty members from institutions that have hosted our Anne Klibanski scholars with MGH scholars on topics that overlap both research areas. And we hope that this also creates new connections and collaborations. And the lectures are open to both the MGH community and that of the host institutions. So today we have the pleasure actually to host two speakers. First is Julie Nonnekens from Erasmus University Medical Center from Rotterdam, the Netherlands. And Julie received her Masters of Science in Biotechnology at the Wageningen University in the Netherlands. She then obtained a PhD in cancer biology with a focus on DNA repair mechanism at the University of Toulouse in France. And then she was a postdoc at the Hubrecht Institute in Utrecht in the Netherlands, working on ribosome biogenesis in cancer and longevity. And in 2014, Julie joined the Erasmus Medical Center in Rotterdam and is now an assistant professor and group leader with a joint appointment at the departments of radiology, nuclear medicine, and molecular genetics. And she has started a new research line that bridges the interests of both departments, in which she is using her experience in the field of DNA damage repair mechanism to study the radiation biology of targeted radionuclide anti-cancer treatment in order to ultimately optimize treatment regimen. Julie has received several awards and is principal investigator on research grants. She is the secretary of the Netherlands Society of Radiobiology and chair of the European Working Group in Radiobiology of molecular radionuclide therapy. So Julie, welcome. And then we have Kerry Reynolds here from the MGH. Kerry is a physician at the MGH Cancer Center and assistant professor of medicine at Harvard Medical School. She currently serves as a director of the Severe Immunotherapy Complication Service and is also the clinical director of the Inpatient Cancer Services at the MGH Cancer Center. Kerry specializes in the care of hospitalized patients, and under her leadership, the MGH Cancer Center's inpatient program evaluates and treats over 5,000 patients each year and serves as a core training experience for Harvard Medical School students, residents, and fellows each year. Kerry completed her residency and chief residency at the MGH, followed by a fellowship in oncology at the Dana-Farber Cancer Center. He started the Severe Immunotherapy Complication Service in 2017 and presently focuses on efforts to uncover predictors for those patients who are at highest risk for adverse events associated with immunotherapies. And Kerry is also the lead editor for Facing Immunotherapy, a book for patients and caregivers to guide them through their treatment journey. So it's my great pleasure to now have Julie, who will start with the first part of the lecture.
Thank you. I will share my screen.
Perfect.
So good afternoon or evening, depending where you are. First of all, I would like to thank for the opportunity to talk about our data here today. It's really exciting to be part of this visiting lecture series. And today I will give a presentation together with Dr. Reynolds. And we named our joint presentation the Black Box of Cancer Therapy. So I'm Julie Nonnekens, and I'm an assistant professor and group leader at the Erasmus Medical Center in Rotterdam, the Netherlands. I have a joint appointment at the Department of Molecular Genetics and Radiology and Nuclear Medicine. And today I will like to talk about the radiobiology of radionuclide therapy. So I thought maybe I'll show you a bit where you can actually find us. So here there's a pin in Rotterdam in the Netherlands. It's close to Amsterdam, which you might know a bit more about. Our hospital, here you can see a picture when it was built a few years ago. At least the extension. We have about 16,000 employees with almost 500,000 square meters in total space. And in 2019, we had about 120 ICU beds, which has almost tripled at the current pandemic. And we have quite a bit of outpatient and inpatient stays. And I think most importantly, all patients are enrolled in clinical research, even for tissues or data, etc. Rotterdam is a city of approximately 1 million inhabitants, and we have a catchment area of 4 million inhabitants. We have quite a lot of medical specialists, residents, and also scientific staff. And not to forget, we are the biggest academic hospital in the Netherlands. So we have an annual budget of almost 2 billion euros. So I guess that's a bit over $2 billion. And about 300 million euros for education and research. So with that, I would like to go into the fun part, I think the research. So I would like to introduce the topic of theranostics to you. I'm not sure if everyone is familiar with this. It's a way of targeting tumors with via tumor-specific receptors with a targeting molecule. That can be a peptide, an antibody, small molecule inhibitors. These targeting molecules can be radio-labeled, And depending on the type of radioisotope, this can be used for diagnostics, for example, for PET or SPECT imaging. You might know the FDG PET that is used for staging cancer patients. And you can also label it with a therapeutic radionuclide, for example, lutetium-177, which is a beta emitter, or actinium-225, which is an alpha emitter. And then it's called radionuclide therapy. And that is the part I will focus on today. So radionuclide therapy, it's working very well. Their patients can really greatly benefit from the treatment. However, we can always do better. And I think it's very powerful to really understand what's happening in the tumor during the treatment. And if we know this, we can also improve the treatment. So that's a bit what my lab is working on. So we are trying to understand tumor responses at the cellular level, but also at the whole organism level, and also measure normal tissue toxicity. And then if you're looking into ways of improving the treatment, there are, I think, 1,000 ways of doing this. But for example, you can change the radionuclide, you can make a combination treatment based on the data that you have, or you can change the peptide. There are different ways of doing this. But this is sort of the black box of radionuclide therapy that I want to talk about today. So I have some examples of our lab that were recently published or quite recent data, just to illustrate the things that we can do. So first, why do we need biology to understand radionuclide therapy? Basically what we do, we give radiation to the tumor cells, they die. If it would be this simple, we are done. That's it. But of course, not that simple. We want to have a maximum efficacy with a certain dose. So what we want to do is to predict the dose upfront, so you know the efficacy. But the dose, if you want to predict the dose, you also have to predict the anti-tumor effects and to predict the side effects. And for this, biology comes into place, because not every cancer cell is the same, not every normal organ reacts the same. For example, you can think of biological characteristics that can influence these effects are cell intrinsic radiosensitivity. Every cell reacts different to radiation. The tumor microenvironment can have many effects on this. For example, the immune response, and that's also something Dr. Reynolds will talk about later on. And there's a lot of heterogeneity of tumor cells for different receptor expression, for example, which I'll come back to later. So these are all things I have to take into account. And we are trying to tackle this in my lab with different methods. So we're doing, for example, in vitro assays where we can do large screenings, where we can find new combinatory treatments. We can do analysis of DNA damage. And we recently purchased a confocal microscope where we can do live cell imaging of radioactive compounds. So there we can do live cell analysis of what's going on in the cell. We also do animal experiments where we can do SPECT imaging, for example, or look at the efficacy of a treatment. And we also do modeling to do better dose prediction models. So we use then the biological inputs to make better computational models for the dose distributions. So I would like to start with an example of a study that we recently published about peptide receptor radionuclide therapy, which has been marketed as Lutathera by Novartis. Patients with gastrointestinal pancreatic neuroendocrine tumors, they respond very well to this treatment. Good tumor response rate, they have an extended progression-free survival, and there's a huge improvement of quality of life for the patients. And this is an example of a patient with liver metastases. Here you can see it on a SPECT scan after different cycles of PRRT. You can clearly see that after the 4th cycle, the tumors are much smaller than after the 1st cycle. So this is an example of a very positive response. However, most patients, we only have a partial response. They cannot be cured at this stage of disease. And it's not possible to simply increase the dosage, because this would lead to normal tissue toxicity, especially in the kidneys due to the clearance, and the bone marrow due to circulation of the radioactivity. So we need to find a solution. But before we can come to this solution, of course, and there might be not one solution, but there might be many, we have to know more about the biology. So we did an animal study where we injected mice with tumors with the radionuclide therapy, and then did scanning, biodistributions, and histology at different time points after treatment. So I will show you part of the data from this study. So what we observed in this study is when we were looking at DNA damage induction and cell death at the different time points, is that there is a lot of cell death induction that you can see here in green. In this picture in blue, there are the living cells, and in green are the cells that are dying due to the treatment. But what you can appreciate here is that it's not homogeneous. There's a lot of heterogeneity in this tumor. And this is also if you would quantify this in all tumors. So after a certain amount, after therapy, there's induction of cell death, and then dying cells are removed. But there's a lot of heterogeneity, and this sparked our interest. Like, how is this caused? So we also measured the level of DNA damage induction, because I already said there's radioactive decay which causes DNA damage induction. And this can be visualized by different markers. For example, the proteins 53BP1 and phosphorylation of histone 2A. And those can be seen then as foci in a nucleus of a cell. So every dot here is one DNA break. And this can be quantified over time as well. And if you would look at the two markers, and here you can also see that there's a large spread of DNA damage, meaning that one cell has many more breaks than the other cell. Well, theoretically, especially in an animal model, you would assume a bit more homogeneous response. But apparently it's not the case. Then we looked into why is this happening? And we looked at different factors. For example, vascularization. Well, that was not it. We looked at proliferation. Maybe high proliferating areas would be more susceptible. Also not the case. But then we looked into the receptor expression, because we are targeting the somatostatin receptor with this treatment. And when we looked at the stainings, you can clearly see that there's heterogeneous distribution. So not all cells have the same level of receptor expression. Then we zoomed into high and low expressing areas, and also looked at DNA damage. It's a bit difficult to see these pictures, but in green you can see the DNA damage, and in red you can see the receptor expression. So we've quantified this. So in these mice, in the tumors that were treated in mice, and what we can clearly see here is that cells that have a high expressing receptor, they have more DNA damage than cells that have low expressing receptor. So this really indicates indeed that the higher your receptor is expressed, the more DNA damage is induced. And we could also see this in vitro in cells, where you can also see in cells that have more receptor expressing, there's more DNA damage. So why is this important? If we look at the animals that were sacrificed at different time points, here you see a large field of view of mice that were not treated, so tumors from mice not treated. There's a lot of heterogeneity. And when you look at 5 and 11 days after treatment, you can see that the red color is going down. That means that the high expressing cells are disappearing. And also when you quantify this, this is the case. So that means either they lose the receptor or they are dying off. And this is really important if you want to predict the treatment response, because it might be that patients with higher expressing cells will benefit more from the treatment. At least that might be one of the factors. And for now, it's mostly if patients have the receptor known from a scan, they will get the treatment, but it's not really evaluated the level of expression. And we've also measured different patients. These are just 2 examples to show you already the difference between patients. Patient 1 here has a very homogeneous level of expression, while patient 2 has much lower and also heterogeneous level of receptor expression. So this indicates the clinical value of these sort of assays. So I'm coming back to this slide that I've already shown before. So this study that I just showed is not a solution on how to improve treatment, but it can be an indication for the receptor expression. But something else that we looked into is, because we know much about the DNA damage, is combination treatment for this. We know the radionuclide therapy makes DNA damage. And if you put it simple, there are single and double strand breaks induced by the treatment. However, our body can repair these DNA damages and thereby counteract treatment. Especially the single strand breaks are very easily repaired and are therefore not cytotoxic. Double-strand breaks are more difficult to repair, and if you have enough double-strand breaks, this would induce cell death. So we thought, PARP is one of the enzymes involved in single-strand break repair. And we thought if we would block the DNA repair, this could increase the number of DNA breaks. And especially if you would block PARP, single-strand breaks are not repaired anymore. And if a cell goes through replication, so cell division, the single-strand break will be converted into a double-strand break. And more double-strand breaks would equal more cell death. And since this therapy really specifically functions on the tumor cells, so there's no irradiation around it, you can really specifically increase the PRT efficacy in the tumors. So this was a working hypothesis quite some years ago. And we've done a lot of different experiments. So this will be a summary of a few years of work. So we used cells to show that if you have the combination treatment of lutetium dotatate with a PARP inhibitor, we have more cell death. So that already showed the work. And we also indeed see there's more apoptosis at 3 days after treatment. The hypothesis was that this is caused by more double-strand breaks. So what we did, we measured also the double-strand breaks. Here you can see them as the nuclear foci. And indeed, in the combination treatment, over time, we have many more double-strand breaks compared to the monotherapy. So this confirmed our hypothesis. And one other observation we had is that also these combination treatments increase the genomic instability. And you can see that here in cells where you see those weird nuclear buds. Those are called micronuclei. And those are the part of the DNA that's come outside of the nucleus because the DNA is broken. So that's also a measure. And we can indeed see here, this is much more frequent in the combination treatment. So we've used different ways to confirm this. But of course, the most optimal one of doing this sort of analyses in the lab is doing an in vivo experiment. So this was really exciting that we could see this in a model. It's really synergistic. It's what we also saw in vitro. And the next step is that we're going to do a clinical trial, phase 1 trial, and that should start this year. So it's really exciting that we can translate something from no knowledge about DNA repair towards the patient. Then switching gears a bit, I want to show some data that we recently published about another type of radionuclide therapy, which is PSMA for prostate cancer. PSMA is frequently overexpressed on prostate cancer, and it's also linearly correlated to disease progression. So the more advanced the disease, the higher the PSMA expression is. And already some years ago, different laboratories showed the promise of this PSMA-directed radionuclide therapy. Here you can see a scan of a patient several months apart. So in the first scan, you can see massive metastases. And after 2 cycles or 3 cycles of radionuclide therapy, you can see the burden decreasing a lot. However, what you can also see here on these scans is that there's uptake of the radioactive compound here in the salivary glands and also in the kidneys, meaning there can be toxicity. And as we know, we should avoid toxicity, of course. So we had the idea for this study to compare efficacy and toxicity of 2 compounds for PSMA radionuclide therapy that are now used in the clinic, but they were not compared before. So we do not know which one is the best. So we did different in vitro studies as well, but I'm going to show you in vivo data here to make it a bit quicker. So we compared the trace for PSMA 617 and PSMA INT, who are both used in the clinic. And if we're looking at the SPECT scan of a mouse, 4 hours after injection, you can see accumulation here in the tumor, still a bit in the kidneys and the bladder, but it's quite minimal. And at 8 and 24 hours, you can see very nice accumulation in the tumor. But if we look at the PSMA INT, you can see a completely different image. The tumor uptake seems to be comparable, but there's massive uptake in the kidneys and the bladder. And this also persists over time. So this indicates that even though the traces are quite similar for their structure, the uptake and the retention in the body is completely different, which might have huge impact on the toxicity. So also if we quantify this for the tumor and the kidneys for the two different tracers, you can see a very different view. And then we also like to measure the tumor over kidney ratio, which the higher, the better. You can see here that the PSMA 617 is much more favorable than the PSMA INT. And that's the same if we would do an autoradiography of these organs. So then you cannot just measure the whole organ, but you take a piece of it and you can see that in the kidneys, in the cortex, there's a huge amount of the radioactive compound. Really showing that in this case, the PSMA INT is not favorable. But this is a mouse, and a mouse is not a human. That's always the big point. So next thing we did is to do autoradiography on human organs. And with autoradiography, you have fresh frozen tissue, and you expose this tissue to the radioactive compound, and you look at the amount of binding. So what we did is we had healthy human kidneys, healthy human salivary gland tissue, and we used prostate cancer xenograft as a control, and we compared the two different tracers. So it's a bit easier to look at the quantification, but if we look at the binding of the two different tracers, you can clearly see that there's about a twofold higher binding to the kidneys of the PSMA INT, also to the salivary glands, but the tumor binding is the same. So this really indicates that the PSMA INT is less favorable than the 617. So I think it's really important that these data will also be validated in patients, so we know for sure what's the difference. And that also shows the importance of doing these sort of assays in deciding what will be, maybe for the efficacy will be the same, but the toxicity can have major impact. So with that, I would like, at the end of my talk, I think for the discussion, the major points that I want to bring over is that these effects that we see with radionuclide therapy, they are dependent on multiple biological factors. There can be vascularization, can be maybe hypoxia, other things, and we've shown that also receptor expression can really be important to know. Well, radionuclide therapy can increase the effects with PARP inhibitors, and but we have to show the efficacy in patients, but also toxicity. We cannot neglect that. And when you want to select the best tracer for your patient, you need biological data. And it's not just, I have this compound, I think it works well, I'm gonna use it. I really think it's very important to have these sort of assays to make the right decision for your patient. Well, of course, I didn't do this work alone. I would like to thank everyone in my lab, my departments, and all the people that were involved in the studies throughout the years. Of course, financial support, very important, and the core facilities from the Erasmus MC. With that, I end. Thank you very much for your attention, and again for the opportunity to give this lecture here today, and happy to take any questions.
Thank you so much, Julie. Do you want to hold questions until the end, and then we can do it combined? And then Kerry, you want to take over, and then we will have a combined question answer session.
Yeah, sure, that's fine.
At the audience, you can already start putting questions in the chat, and I will monitor it and ask it at the end.
So good afternoon and good evening. My name is Kerry Reynolds, as they said in the introduction, and I just want to thank Drs. Klibanski, Bradella, and the Center for Faculty Development, because for the scholars to bring together both community and opportunity is truly a pleasure. So thank you. And today we're going to talk about a different black box of immunotherapy. I have no disclosures. So maybe we should frame this about immunotherapy or immune checkpoint inhibitor in cancer. Where are we now? So if we go back to 2011, and if you were diagnosed with melanoma, and it was metastatic to the local lymph nodes, bulky lymph nodes, unfortunately, the overall survival was on average about 12 months. And if it was with lung metastasis, that survival was as low as 8 months, or with liver, bone, or brain, down to 4 months. We had tried multiple chemotherapies for decades, and the standard of care was dacarbazine. It only had a response rate of about 5 to 20% and only lasted for months on average. It walks in the immune checkpoint inhibitors, and just less than a decade later, the entire field is revolutionized. So melanoma in 2020, recent data of 4-year overall survival data from the first immune checkpoint inhibitor approved, ipilimumab, showed that 34% were living. In addition, the 5-year overall survival for PD-1, which we'll also talk about, had an over 40% that had these durable, lasting responses. So it's really revolutionary, and I want to make sure today we leave with the fact that immune checkpoint inhibitors are a good news story. So how do these agents work? So this is a basic graphic, and really the top is just showing that green cell is an antigen presenting cell. And many times throughout the day, right, your immune system is really designed to determine self from non-self. And so that little blue antigen there is presented on the MHC molecule of an antigen presenting cell up to the TCR of the T cell. That is signal 1. But the immune system is tightly regulated. Activation actually requires a second signal, a co-stimulatory signal. So there you see in red, it's CD80 and CD28, and that second signal is key for activation. But it's a tightly regulated system in that we also regulate this response with checkpoints. So soon CTLA4 goes to the cell surface, and the CTLA4 binding actually outcompetes the second co-stimulatory molecule. And that's the thought around central tolerance. Now, out in the periphery, there are checkpoints or breaks as well. So here you can see this in the middle, it says trafficking of the T cells to the peripheral tissues. Because once they're activated, you have proliferation, you have cytokine release, and they start to migrate to the tissues. And once there are also balances or checkpoints in place. The tissue upregulates PD-L1 or PD-L2, and you have PD-1 on the T cells, again, to have breaks of the system. But cancer can co-opt this and evade the immune system. And so these drugs that we're talking about are monoclonal antibodies to the checkpoints. So they activate the system. The first approved was ipilimumab, an anti-CTLA4 agent, and then you can see 6 others have since been approved. 3 against PD-L1, atezolizumab, durvalumab, and avelumab, and 3 against PD-1, nivolumab, pembrolizumab, and cemiplimab. So these are the agents that we are going to talk about today. And you do not have to read the fine print here. It's much more to take a step back and just see where the field has changed. Since that first approval in 2011, we now have over 50 indications approved in cancer for immune checkpoint inhibitors. It's really remarkable. Approvals of those 7 drugs in over 15 tumor types. And importantly, and really interestingly, where these darker blue boxes show that we're starting to approve more combination therapy. So whether that's immune checkpoint inhibitor and another checkpoint inhibitor, or immune checkpoint inhibitor or target therapy, or immune checkpoint inhibitor and a chemotherapy, but you can imagine that changes the presentation of patients as well. But the reason why these approvals are so exciting for the field is we're seeing these responses. You can see now not every type of disease has the response we see in, say, melanoma or Hodgkin's, but it is present and it is here to stay. So I think it's really remarkable. The issue, though, is that the immune system was tightly regulated to prevent ongoing autoimmune-like conditions. And when we dysregulate this in a subset of patients, we can start to see adverse events or immune-related adverse events, IRAEs. Again, on the left, this is just something we put together that really showed some of the cases that we see, but it's too fine print. So on the right shows you the systems that are often and can be affected. So that's the GI tract with colitis, or the liver with hepatitis, or the lungs with pneumonitis. You can get endocrinopathies, anything from hypophysitis, thyroiditis, adrenalitis. It can affect the kidney, the marrow. It can affect the joints, the skin, the nerves, the eyes, you name it. So it really is a wide array of organ systems that are involved. And that doesn't really give you the whole sense. And I don't get to go into this case in detail, but I really wanted to share it with you. This is the case of a 47-year-old gentleman who wanted us to share his story. So he was 2016, and he was diagnosed with cholangiocarcinoma that was metastatic. He started on chemotherapy with a round of gemcitabine and cisplatin for several months, but unfortunately progressed. That's when he had a combination therapy of an immune checkpoint inhibitor and a targeted therapy to go after his disease. But that was only in November. In January of that next year, he presented with profound hypoxia, so a low oxygen level, infiltrates, and an ejection fraction of 45%. Now, sometimes when we see those lung infiltrates, that could be anything from, infection to fluid to even inflammation of the lungs. But he was diagnosed with an elevated troponin in a biopsy with myocarditis, inflammation of the heart, second to nivolumab. He did well with steroids alone. And in fact, really exciting because cholangiocarcinoma is very difficult to treat. He had a decrease in the size of his liver lesion. However, subsequently, March of that year, he came back into the hospital and he was diagnosed with a second immune-related adverse event of hepatitis, and it didn't stop there. The immune system was just relentless. He had marrow toxicities 3 months later, thought also to be immune-related. And this whole time he had been on immunosuppression because that's how we try to control the immune activation. Unfortunately, so bad that he's been on so much steroid, he had extensive compression fractures from chronic high-dose steroids, needed pain management and kyphoplasty. And we never got control of his immune system. He passed away from refractory toxicities related to immune checkpoint inhibitors and gave us the indication we really needed to organize in a different way to care for these patients and to tackle these new set of diseases that we were seeing. So as part of that, one of the first part is really to be able to speak the same language about what we're seeing. And the common terminology criteria for adverse events, it's online, it's available to all, and it helps us because it grades the toxicity. It's either mild, grade 1, moderate, grade 2. I kind of put it like a stop sign here. So you get green, go, yellow, we have to really take a bit more caution, and then red or the more severe toxicities are grade 3 and 4. For example, if someone presents with diarrhea and it's less than 4 stools above baseline, that's still grade 1. But if it gets into the 4 to 6, that's more moderate, or over 7 stools above baseline, that gets into the severe category, and so on. It just, I think, gives you a framework then to think about how often do these occur, right? How common is this with these drugs in terms of the autoimmune toxicities? On the left here, I just wrote it out by different regimens. So PD-1 alone, or CTLA-4, combination 2 checkpoints together, or combination checkpoint and chemotherapy. And you can see if we take any grade toxicity, it's not a small number. It can be anywhere from 74 to 90%, but thankfully these are often mild. The severe happen much less likely. You can see the percentages there, anywhere from about 14%, but when you start combining, the combinations can get to 40 to 50% of toxicity. And I like that column on the right because it really shows how many times is it severe enough to need to stop treatment. But you can also see these are rarely single-digit numbers. Thankfully, fatalities are even more rare. So with PD-1, it's less than 1%. And you can see it goes upwards as high as to about 1.2% with the combination therapy. But these are fatalities not related to their cancer. It's specifically related to these autoimmune events, similar to the case that I showed you. And how do we manage these? And this is what is tricky because a lot of this is expert opinion, not evidence-based at the moment. So the persistent or mild ones, that grade 1, often we can treat symptomatically, but we can keep using the immune checkpoint inhibitors. Whereas the moderate, it depends on what toxicity you have, but often we need to start oral corticosteroids steroids with prednisone, a mg per kg or so a day. Now, the exception to this are the endocrine ones, because for hypophysitis, for thyroiditis, you can replace the actual hormone and we don't need the immunosuppression, but for most others we do. And then when we get into that more grade 3 or 4 category, we often have to even hospitalize these patients using high dose of IV steroids. And then if they don't get better at, say, hour 48 or 72, we have to start thinking about escalating immunosuppression. And similarly, we are borrowing from other autoimmune-like conditions at this point because we don't know enough about these specific diseases. But you can see we start things like Cellcept in the case of hepatitis, or infliximab in the case of colitis, because we're borrowing from the autoimmune literature. So this is just a summary to kind of frame it, that now we know immune checkpoint inhibitors are standard of care. We know the success of single agent immune checkpoint led to more combination approvals. We know that this is a good news story because a subset of these patients experience a durable long-term response, and that's what every cancer patient needs. And we need to be aware of these unique autoimmune toxicities. They often occur early within the first 3 to 6 months, but unlike chemotherapy, they can last for weeks or months, and they can even occur after the immune checkpoint inhibitor is stopped. I hope I conveyed that the mechanism is not well understood, because that's what we're going to try to tackle in the rest of this presentation. But the immune-related adverse events, they're typically low grade, but severe events can happen, and even fatalities. And after the case of that 47-year-old patient and several in that year, we really had to take a step back because there were so many questions and so few answers. How do we communicate the possibility of this kind of toxicity to our patients? What are best practices to manage these atypical presentations, right? He started with one, he developed 2 others, and they had significant implications. How do we prepare our providers or assemble a team? Because we need a lot of specialty expertise. Can we uncover predictors? Why is it that he developed such significant toxicity, and what are we treating? What is the underlying pathophysiology of these individual conditions, and can that help us inform our treatment of toxicity? And can we uncover biomarkers that indicate if a patient is just going to do well with a small dose of steroids alone, or if they're going to have a complicated one? So many questions, few answers. And that's when I kind of circled out, what all would we need if we were to develop a team like this? And if you think about it, the oncology expertise has to be, in all different types of cancer care. In addition, the subspecialists are critical across, say, cardiology, GI, pulmonary, and so on. And so we developed the Severe Immunotherapy Complication Service. This was in 2017. And since that time, we have actually seen all the patients together in terms of as a team on the inpatient service, and outpatient subspecialty clinics have been set up. There are now 51 members. They cross a lot of different departments and divisions at our institution in order to get the right expertise. And 15 of the members are bridging between the clinical care or clinical research and the lab. The individual shown at the top right is my partner in crime, Dr. Chloe Villani, who runs our single cell genomics center here at MGH. And you can see the individuals in her lab that are really training both on the clinical side and on the translational side. So 1, we care for patients. 2, we had to develop a clinical registry in order to really understand the presentation and the phenotype of these individual conditions. And 3, it's the translation to make sure that the samples are getting to the lab. And so what we really kind of tried to go over from 2011 even up to the late of 2018 to see how many cases are we seeing. Here it shows in blue that the cases are increasing year by year, but importantly, the rate of admission is actually somewhat going down. It's just that we're treating so many more patients. So more of them are being able to treat without admission, but it's just that we're increasing because of sheer number of patients that are treated. And similarly on the right, it just shows that the yearly change in proportion of the patients that we're admitting were also immune-related adverse events. This is really showing the type of condition. So whether it's a gastrointestinal or dermatological, pulmonary, each of them have a color. And the only thing this is here to show is that it's a little bit of everything. So you really do have to have a wide breadth of expertise. So this section was more to say that serious immune-related adverse events, they can involve a wide range of organ systems. There's recognition across the board that disease-specific subspecialists are essential. It's critical to develop expertise to recognize these immune-related adverse events and educate all types of providers, whether that's MDs, nurse practitioners, nurses, residents, patients, you name it. And the Massachusetts General Hospital has a highly specialized severe immunotherapy complications, clinical and translational research effort. And Rodrigo asked that I share his case, his name, and I'll also give you a quote from him later, but inspired by him and the patients with severe immune-related adverse events. So together as a group, the individuals, these are the papers that we have really tried to just define what we're seeing and what the clinical predictors are. So that has been key in the effort, as well as when Julie said that everybody in the hospital is part of clinical research, I thought, what a fantastic model to be able to move the field forward. It's really incredible. Here, as part of this service, we ask each individual patient, we are thinking about who would be willing to consent for blood and tissue at the same time. It's not standardized across the board, but in the last several years, we've collected over 250 patients because it's critical to have the blood and the tissue to be able to understand. And so what they're doing in the lab, say for example, it is a liver biopsy that was taken to determine if a patient had hepatitis. If the patient consented, an additional sample is also sent for research. This tissue is dissociated into single cells and separated out. You have the immune cells separated out from the blood cells, and then you can really start to isolate the single cells, looking at single cell RNA sequencing, grouping those expression patterns together so that you can see how does the healthy state compare. And luckily, Dr. Vellani is also working on the immune atlas, so we have a sense of the healthy state in these organs. And then the disease state on the right. So do you see how there's these groups of cells? So that's exactly what we're trying to do, is pull out what is different in the disease state. Could that be targeted? Importantly, if you find it in the tissue, can you also find it in the blood? Because we have no true diagnostics or biomarkers. So since we've started, we have collected across organ systems, and I think that will help us understand how they relate to one another. An example of this is just the patient below that was a renal cell patient that had ipi-nivo, so that is the CTLA checkpoint, and the PD-1, so it combination therapy. Again, remember, we recall that 55% of those patients can develop severe toxicity, and unfortunately, he did. And he had multiple toxicities. So he had a myocarditis, he had a nephritis, and a myositis. And so this shows that the myocardial sample off the bat, we were able to send to the lab along with paired blood. And then we try to get blood samples at the time of their recovery. In addition, when they develop a new immune-related adverse event, like when he developed the nephritis and the myositis, and collecting blood after that as well to really longitudinally map these patients and understand the dynamics over time. So this is really showing, again, much of this work. This work is unpublished and it will be coming out with much more detail, but just to get a sense of what they're really working on. So if we look at the first 136,000 cells across our 37 samples, when we think about colitis, so colon samples, arthritis, so synovial fluid, hepatitis, so liver biopsy. This is showing on the left a single cell plot. Remember, we are grouping these cells together. So you can see how the T cells are in one area, the plasma cells, the myeloid, and trying to get a picture of what's present. So if we actually look at this and we look at across the samples that we have, so these are the first 37,6 samples, what I hope you take away from this picture on the right, besides it's quite beautiful, is that all the colors somewhat mix. We don't see how it's all pink or all blue. Instead, they're actually quite mixed. indicating that there are probably shared biological programs driving these events, and that it's not necessarily patient specific. And I think that is important to us, and it's going to be important for therapeutics as we start to think about that. There's many other analysis ongoing. But the reason I wanted to go back to Rodrigo is because before he passed, about 3 weeks before he passed, I was able to call him and tell him, you know what, we're going to set up this service. We're going to bring a bunch of people together, and we're also going to try to bring science to sit side by side with us. And he heard about this. He was really ill. He was actually even hospitalized at the time. And he said, what you are going to do is going to benefit so many. I'll be happy and honored to help in any way possible to make this journey easier for others, because that will make mine even more worthy. And I just won't forget those words. And then his wife has been with us, you know, through many presentations and is really involved in the entire service. But she said, I feel exactly the same. If our journey helps someone else's be less painful, then it's more worthy. If awareness was created, then what we endured has more meaning. This is how science advances. This is how we learn. This is how medicine writes the new chapter. I just hope God gives me enough years to witness the progresses that I am sure the MGH team will achieve. He will remain alive in that progress. And one day my kids will have one more reason to even be more proud of their daddy. So I think it just goes to show, if that's not inspiring to keep going, I don't know what is. And it's thank you to our patients, thank you to our families, and the fact that it really does take an army, many of who are listed here, at least in our own institution. But we have a lot to learn, and we are going to pursue this in many different directions. So I look forward to any questions that you guys may have.
Thank you so much, Kerry and Julie. And this was just so powerful, Kerry, this last statement. So thank you. We can take it off sharing screen so we can all see each other. If there are any questions from the audience, unfortunately, this is a webinar, so I cannot just unmute yourself, but please put them in the chat and I will ask them. So I have a couple of questions. This is so fascinating, and I'm a radiologist. I didn't say this at the beginning. So Julie, it's very fascinating what you presented. Kerry, you said that the side effects are, well, they're really the problem, and you don't know who will develop them and who will not. Is there any way, imaging-wise, Julie, maybe for you, to figure out who will, are there any new tracers or markers who will maybe get more cardiac side effects or renal side effects, or is there anything how you could assist each other?
Yeah, well, thanks, of course, for the compliment on the talk. I must say I'm not a specialist on the imaging part per se, but I know in the nuclear medicine field, many different tracers are in development or being tested in the clinic for imaging of inflammation, immune responses. So yes, I definitely think that there can be a way for nuclear medicine to be part of this immunotherapy discovery or predicting side effects or efficacy, etc. So yeah.
And when Julie and I met before to think about our talks, we were even talking in our own group. People are working on a variety of different tracers, even thinking about granzyme B, because that can be released in cytotoxic T cell activity. The issue is that it's going to be hard to know what is inflammation versus what is true pathology, immune-related adverse event versus what is tumor response. And so I think we have a long way to go to make sure that something is, you know, specific enough. But we hope to work, you know, in many different avenues.
You can collaborate and find out something. I just want to say Gabriel Christine is here. He is my mentor for in another society, who says very nice presentation. Thank you so much to both presenters. So Gabriel, thank you for being here and making this connection. He's at Erasmus University in Rotterdam. Julie's here. There is another question here. Super talk, Dr. Reynolds. And when can you predict patient with high probability of toxicities? What will you change in the clinical practice? Consider a different therapy, and if any available?
Laura, this is such an important question because also we have to put it in the context of melanoma. When I showed that first slide in 2011, you're talking about when it's widely metastatic, we have an overall survival, if we do nothing, of less than a year, even months at times. And so you kind of have to put it into the context. But where I think this is really important and why I think we need to do it now is that we are starting to use immunotherapy much earlier. So not the people that have no other options, but for example, even in breast cancer, we're starting to think about using it early in the disease where it's potentially curable, but we can get a higher pathological complete response if we're using immunotherapy. So in that case, when it's only for us, a marginal benefit, if we knew that someone was that high risk, I think it will be very important to think about, should we be using it in the curative setting? Secondly, I think we could monitor those patients somewhat differently. Third, we could maybe avoid combination therapy in those patients and think more about single agent. So there's a lot of clinical practice implications, but you're exactly right. Even if someone had metastatic melanoma with a really high risk, it's unlikely that we're going to potentially withhold all immunotherapy throughout the course of their disease, given the horrible prognosis. But I think it's a work in process, but the more knowledge we have, the more tailoring or personalized we can be with the regimens.
Great. I have another question out of personal research interest. Walter Longo, he has done a lot of studies on fasting that before to decrease the side effects of chemotherapy. He has done amazing experiments in mice where he had them fast for 24 hours before giving high dose chemotherapy, and the mice who fasted, they survived, and the one who didn't, they died. There are some studies in humans, in breast cancer especially. Is there any research or are there any studies that looking at pre-treatment fasting to decrease the side effects from immunotherapy?
It's such a good question. To date, there are none. I think it's just so difficult and challenging to really predict who is going to have these side effects and how do we mediate that. And I think it's all a work in progress. I think many additional studies about what we should be doing before someone has it is going to really help us then refine it. But so far to date, fasting hasn't been connected.
Great, thank you. It is 1 o'clock. I would like to thank the speakers, especially Kerry and Julie. It was an absolutely fantastic presentation. And I also would like to thank the audience for tuning in here. So we have had a great number of people, and especially I saw some Dutch names. So I hope we had some Dutch audience, people from the Netherlands. And thank you so much for spending this hour, and thank you, and I'm looking forward to the next lecture.